Hello, very good evening everyone. This is your Shruthi Maam and today I am so happy to be doing two very important units for our exam. Okay, so firstly we're going to do reproduction and second of all we're going to do human welfare. Both of these units are super important and both of these units are going to add a lot of marks to your entire exam. Okay, now if you look at the entire NEET paper, reproduction has 3 chapters, one of which is botany, sexual reproduction in plants, and then the other 2, reproduction in humans as well as reproductive health, come under the zoology section. Now all 3 chapters put together, easily you will be getting close to 6 to 7 questions, of which reproductive health alone can carry almost 3 questions. Okay, so roughly 7 to 8 questions you can expect from this section. So roughly 30 to 35 marks you can expect from just reproduction unit. Okay, now apart from that we have human welfare. Now human welfare consists of 2 chapters. One is human health and disease, the other one is microbes and human welfare. Now if you look at human health and disease, there majorly you will be getting questions from the diseases, very straightforward questions, AIDS, cancer as well as drugs. So these are the most important parts of human health and disease. And when you look at microbes and human welfare, there it is super straightforward questions that you will be getting. Now you just need to learn the microbes, what they contribute in and what we are using that particular product for. Now if this particular segment is clear to you, then human welfare, the entire 2 units put together, 2 chapters put together, you can again expect about 5 to 6 questions. So today's class alone can give you roughly say 10 to 12 questions in your paper. Okay, which is what 10 to 12 questions corresponds to almost 40 to 48 questions marks in your NEET paper. Okay, so all you need to do is follow me through the class. Do subscribe the channel guys, because every single day I'm going to be coming with content to help you for your NEET exam, for your future choices, counseling, everything we will be taking care of. Okay, is that clear? So healer, don't worry, we will try to wind up at the earliest. Now remember that my class is a no nonsense class. I'm not here to discuss anything that's irrelevant. I'm not here to waste your time. So I'm only going to focus on what is important, and I'm also going to write the notes in a way that you can use for your last minute revision. Okay, so all I want you all to do is definitely subscribe and join my Telegram channel where I'll be sharing all these notes for free. You can also try out my AI tool guys. This is a free tool that you can use to practice infinite mock tests and also do infinite PYQ practice. Okay, is that clear? So subscribe, like the video, and let's start off with today's class. Okay, so today we will start with reproduction. Okay, I'll go with the first 3 chapters, then we will go for the human welfare. Okay, so firstly, coming to the first chapter, which is sexual reproduction in flowering plants. Okay, now when I say sexual reproduction, remember that reproduction can be sexual or asexual in most plants. Okay, where asexual involves only one parent and the genetic components remain the same. In sexual reproduction, definitely a bit of variations are produced, and this happens only in the flowering plants. Okay, so when I say sexual reproduction in flowering plants, the first thing I need to know is the flower. Okay, so what are the different parts of the flower is the most important first segment that we need to understand. Okay, so now when you look at a flower, okay, we have the first part which is called, okay, not exactly the first part, but I would say the central piece called as the gynoecium. Okay, then we have the androecium. Okay, now after the androecium we have the corolla or the petals, and then we have the calyx or the sepals. Okay, so now these are the sepals. The main function of the sepal is to protect the plant, okay, and give a lot of support and lot of protection while the flower is still a bud. And then we have the petals. Okay, so sepals make up the calyx, okay, and the petals make up the corolla. Now, both of these are unwanted in the sense they don't directly contribute to any growth or reproduction or anything. They just auxiliary elements. Some plants may choose to not even invest on these extra parts at all. Okay, but when you look at the center two parts that we just spoke about, one is the gynoecium and the other one is the androecium. Both of these words are super important. Now, androecium is the male part of the flower and gynoecium is the female part of the flower. Okay. Now, when you look into the details of it, when you look into the androecium, what we have in the androecium is filament and anther. Okay, so this is the filament and the structure on top is the anther. Now the anther has two lobes, so that's why we call it as a bilobed. Okay, and if you look inside these lobes, we have two sacs each. Okay, so they are dithecous, means there are two sacs in each lobe. Okay, now when you look at the ultrastructure inside the anther, okay, what we have is a tissue like this inside the anther. Okay, so here we have an outermost epidermis, okay, and then we have an inside endothecium, okay, then we have some middle layers, okay, that are present, and inside the middle layers we have the microsporangium. Okay, so we call it as the microsporangium. Okay, now this microsporangium is nothing but the sporogenous tissue that we are talking about. But this sporogenous tissue is covered by another layer of cells, okay, called as the tapetum, which is basically there for nourishment and support of this sporogenous tissue. Okay, clear to everybody? Now, when you look at the microsporangium inside, okay, now definitely there is a microspore mother cell, okay. Now, this microspore mother cell is going to further divide, okay, which is 2 in number, is going to undergo meiosis and is going to form 4 haploid microspores. Okay, now if you look at the structure of these microspores, what happens is this particular cell further divides and forms what we call as a vegetative cell, okay, and a spindle shaped generative cell. Now, in this generative cell, we've got one nuclei, and this nuclei further divides into 2 male gametes. Got it? Now, these microspores are nothing but develop into the pollen grain, and inside the pollen grain, we will have 2 male gametes. Is that clear to all of you? Everybody? That's about it. Okay? Good aesthetic. Okay, so aesthetic, first what happens is we have a microspore mother cell who will first differentiate through mitosis. That's all. Okay, then after that we will have meiosis. So meiosis is a 2-step process. We'll have meiosis 1 and then meiosis 2, and at the end of meiosis 2 we will be getting the 4 haploid microspores. Okay, healer, microspore is the last cell that is going to differentiate into this pollen grain. Okay, microsporangia is the starting tissue, the sporogenous tissue which is giving rise to the microspore. Is that clear, healer? Aesthetic? Clear to all of you, everyone? Perfect. Okay, now this is about your androecium. Now when you look at the gynoecium, Okay, when you look at the gynoecium, first we have a landing place called as the stigma. Okay, now inside the stigma we have a long tube which is called as a style. Okay, inside the style we have the ovary, and inside the ovary we have the ovule. Okay, now this ovule in its fact is the actual region where my megasporangium is present. Okay, where my megasporangium is present. Okay, now in the megasporangium as well, there will be the megaspore mother cell. Okay, now this megaspore mother cell undergoes meiosis and it produces 4 haploid cells. But in that, 3 will degenerate. And only 1 functional megaspore is present. Now this one functional megaspore, what it happens is this particular cell will now divide into 2, into 4. Okay, now these 4 cells will again become 8 cells, but here remember that it's not cell. This one functional megaspore will be in one entire cell, but inside the cell the nucleus will start to divide. Okay, so what essentially happens is this one functional megaspore nucleus will now start dividing. So it will become, 1 will become 2, 2 will become 4, 4 will become 8. Okay, and once it is 8 nucleate, they will start assembling themselves in the cell. So if you see, we have one chalazal end and one micropylar end. Okay, now towards the chalazal end we will have 3 antipodal cells. Okay, so we'll have 3 antipodal cells, and then towards the micropylar end we will have 2 synergids and 1 egg cell. Okay, is that clear? So we will have 1 egg cell and 2 synergids that are present in the micropylar end, and then in the center, because this whole big thing in itself is the central cell, correct? Now in the central cell we will have 2 free polar nuclei that is present. That's all. This is the mature megaspore ovule that you will see. Is that clear, everybody? Is that clear to all of you? So now next we're going to study about pollination, how exactly this pollen grain is coming and the gametes are able to meet the egg cell. Is that clear? Before that, definitely do subscribe and like the video. Join the Telegram group for all the free notes. Is that clear to all the new students who are coming in? Remember that you will get all the free notes in my Telegram channel. So don't forget to subscribe the channel. Alright? Great. So now we have a egg cell, we have a male gamete that is formed. Now we need both of them to meet. That process is what we call as pollination. Is that clear to all of you? Now when I say pollination, it is nothing but transfer of the pollen grains to the stigma of the flower. Okay. Now when you look at pollination, it can be either autogamy, means it is the same flower. Okay, now the same flower can either be a closed flower, okay, where it is a cleistogamous flower. Okay, so cleistogamous flowers are where it will not open at all. It will remain confined itself. So there is no chance but to actually undergo self-fertilization. There's no other way, because if it does not open at all, I'm not even going to get exposed to anybody else, correct? Now if you look at the chasmogamous flowers, these undergo self-fertilization only, but they open out to the outside. Okay, so they will open. Is that clear, everyone? So healer, On the YouTube channel itself, I'll be discussing questions one by one. Okay, so there I'll do chapter-wise questions. On Telegram, I'm doing a study with me challenge. Okay, so in the study with me challenge, I'm giving you portions for every day's revision and a test link with that. So when you will have this particular chapter, the test link will have questions for it. Okay. Got it, healer? Perfect. Okay. Now, cleistogamous and chasmogamous flowers will undergo only self-fertilization, self-pollination. So, the pollen grains from the androecium of the same flower will pollinate into the stigma of the same flower. Okay. So, there is no variations or no any other possibilities created. Now, the other one is a geitonogamy, where it is a different flower. Okay. But it is the same plant. Meaning, if let's say this is my plant, I have one flower here and I have one flower here. The pollen grain from here will come and fertilize into this. Understood? That is geitonogamy. Now, entirely when it is two different flowers, we will call it as xenogamy. Okay? So, different plants, different flowers. Okay, so this is xenogamy. Is that clear, everybody? Now, when you look at the modes by which this pollination can happen, there is a lot of means. Okay, so who are the agents of pollination? We have hydrophily, which is through water. Okay. Then we have anemophily, which is by wind. Okay. Then we have entomophily, which is by insects. Apart from that, we have ornithophily, which is through birds and so on. Okay. Is that clear, everybody? So, through these different kinds of agents, we will take the pollen grain from the androecium or from the anthers of one plant into the stigma of another plant or another flower. Understood, all of you? Now, once the stigma has received the pollen grains, okay? Now, once the pollen grains have been received, remember that 60% of the flowers, when they are released, they will have only one cell in the generative cell, and then later, after landing onto the stigma, they will divide and produce two gametophytes, okay? Two gametes of the male cell. But most of the time, sometimes when they are released itself, they will divide into the two cell stage and then they will be released. So, it depends. But either way, when they come to the stigma, they are already ready. Okay. Now, there is a pollen pistil interaction that happens here. Okay. And because of the pollen pistil interaction, if the pollen grain is something that is able to, is compatible with this flower, and if the flower is accepting this pollen grain, only then the pollen tube will be growing. Otherwise, it will not generate the pollen tube. Okay, is that clear? So, through this pollen pistil interaction, once the pollen grain is identified to be, okay, this is somebody who's compatible to me, I will pick up this tube and I will come inside. So, here I will have the two male gametes traveling inside the pollen tube and reaching the ovule. Is that clear? Now, before I go to fertilization, which happens in the ovule, I will talk to you about the outbreeding devices. Okay. So, what are the outbreeding devices? If you see, we can prevent the self-pollination from happening by using different kinds of methodologies. That's what we call as these outbreeding devices. Okay. Because if self-pollination is to happen, generation after generation, the self-pollination will lead to a lot of inbreeding depression. I will not have variations and is only going to cause more problem for the plant. Okay. So, in order to prevent that, what I can do, I can have different positions. Okay. If my gynoecium is present above the androecium. The androecium's pollen grains naturally cannot fall on the gynoecium that is on top. Correct. So then what happens? It will fly away and some other pollen grain only will come and fertilize. So like that, I can prevent the self-pollination. The other one is the synchronization in terms of receptibility. What is receptibility? The ability to sense the pollen grains. So the pollen, the androecium and the gynoecium will mature at different times. So when the pollen grains are being released, the stigma is not even receptive to pick up the pollen grains. How will the fertilization happen? So there will be no pollen tube that is created. Okay. So apart from that, there can be barriers. Okay, and there can be many other techniques by which I can prevent the self-pollination also. Okay, so majorly the goal is to prevent self-pollination so that I can have variations and my species can evolve. Understood? Is that clear? Okay, so Kiyomi, what you can do is you can subscribe the channel and come back and watch the live video later. So you will be able to see the video recorded if you're not able to attend the class now. Got it, Kiyomi? Perfect. Okay, then. Now, coming to the next part, which is the fertilization. Okay. Now, in my egg cell, I have synergids and my egg cell that is present. I mean, in my ovule, I mean to say. Then I have two polar nuclei that is present here. And then I have the antipodals. Okay. So total we have we have 7 cells because 7 cells are there but 8 nucleuses are there. So 8 nucleate stage. Okay. Is that clear to all of you? That's it. Okay. Now in this particular case, what's happening is my pollen tube is going to come and it's going to be 2 male gametes. Okay. Now what are the 2 male gametes going to do? One male gamete is going to fuse with the egg cell. Okay. So male gamete plus egg cell is nothing but what I call as syngamy. And this leads to the formation of a zygote. Okay. Then the second gamete is going to go and fuse with the polar nuclei. So male gamete plus the polar nuclei is going to form the primary endosperm nucleus. Okay, so this is going to form the endosperm. So now if you see, the zygote is 2n chromosomes because one egg and one male gamete. But whereas if you look at the polar nuclei, there are already 2 nucleuses there and one fusing from the male gamete. So this becomes a triploid cell and this is the one that is going to give nourishment to the zygote until it is going to germinate in the ground. Okay, is that clear to all of you, everyone? Alright, any questions so far? Any questions so far? So as a result, we have one fertilization that is syngamy, one fertilization that is forming the endosperm nucleus. So altogether, 2 different fertilization processes are happening and that is why we say that double fertilization has happened. Okay. That's it. Now, once the double fertilization is complete, it will enter the post-fertilization events. Okay, so what are the post-fertilization events? My zygote will now mature and form the embryo. The ovule will form the seed. Okay, my ovary will form the fruit. Okay. Apart from that, the petals, sepals, all this will, anthers, all of them will fall off and they will, it will just degenerate like that. Okay, it will just fall off. Is that understood? Clear, everybody? Any questions so far? Now, if you look at this particular embryo, the embryo is nourished by the endosperm until it finds the ground and finds external nutrition to start growing. Okay. Now, this particular embryo, it starts as a single cell stage. Okay, it moves further and it forms a slightly mature embryo, I mean slightly bigger embryo. There is a 2-cell, 4-cell, 8-celled stage. Okay, now after that it forms what we call as a globular embryo, a heart-shaped embryo. Okay, all these stages will form, will cross, and it will form the mature embryo inside the seed. Okay, now when the seed has endosperm that is left, we call it as albuminous seed, or if it is not there, we say a non-albuminous seed. Okay, now a seed has a cotyledon and then the embryo in between. Like I said, when it forms the mature embryo, will have something called the plumule, which will give rise to the shoot, and then we'll have a radicle which will give rise to the roots. Okay, is that clear, everyone? Okay, any other questions? Joe, Randeep, Tarun? No other questions? That's it. So then what happens is, after this we will have the growth. Now once it finds the right space, once it finds the right conditions, the plumule will generate into the shoot, the radicle will generate to the roots. Okay, is that clear, everybody? Now in this chapter you have to learn something called as apomixis and parthenocarpy. Okay, now parthenocarpy is producing fruits without fertilization. Okay, whereas if you see apomixis, it is producing seeds without the fertilization. Okay, now sometimes what happens is the tissues and the nucellus and all that will come inside and start contributing to formation of an embryo, and that is when a polyembryony can take place. Okay, when other tissues close to the ovule will come inside and contribute to the development of the embryo. Okay, like in orange, in any citrus fruits and all, you will see multiple small things inside the seed. Okay, which is nothing but a polyembryony. Clear, everybody? Healer, the synergids and all will disintegrate. Okay, so the synergid is basically part of the egg apparatus and it's only helping to guide the male gamete to reach the egg cell. Okay, healer? Okay, is it clear? Yes, Tarun. All lemon, orange, all citrus fruits will have it. Okay? Perfect, that's it. So we are done with all the important parts of sexual reproduction in flowering plants. And the next one we are going into is human reproduction. Is it clear? Shall we begin, everybody? Don't forget to subscribe and like the video. Okay, I'm going to see how many of y'all are actively watching the class so that I can see the like button go up. Okay, and also guys, please do join my Telegram channel where I'll be doing a study with me challenge. Every day I will share the portions you need to revise. I'll share you a test link to practice as well. You can use my free tool to do n number of mock papers and n number of PYQ practice as well. Okay, everybody? Yes, Tarun, Raghav, we are going to finish all reproduction as well as human welfare chapters. Total 5 chapters we are going to finish today. And we are done with 1 chapter. Okay, shall we go ahead? Joe Benin, I just told you, right? So, there is one fertilization happening between the male gamete and the egg cell, which is nothing but the syngamy. And then the male gamete with the polar nuclei forming the endosperm, which is the triple fusion. Okay, so triple fusion and syngamy together is what we call as double fertilization. Okay, any other questions, anybody? Any other questions? No? Shall we continue on to human reproduction? Perfect. So in human reproduction, if you see, we have clear-cut processes. Okay, so first we have a pre-fertilization event. Okay, which is nothing but anything that happens before the fertilization, including your gametogenesis as well as the insemination for the gametes to meet. Okay, then we have the fertilization process. Okay, after fertilization we have the post-fertilization events. Okay, so the post-fertilization events include implantation, gestation and parturition. What is implantation? Attaching to the mother's uterus. Gestation is the entire pregnancy, and then childbirth is what we call as parturition. Is that clear to all of you? Is that clear to all of you, everyone? Okay, so here, when I look at human reproduction, before we go into the pre-fertilization, fertilization or post-fertilization events, we will look into the anatomy of the male and the female systems. Okay, now when you look at the male reproductive system, we have the primary sex organ, which is nothing but the testis. Okay, so the testis is basically present in the scrotum. Why? To maintain about 2 to 2.5 degrees less than the body temperature. That is what will help in spermatogenesis. So for that, the testis is placed in the scrotum outside of the abdomen. Okay, now inside the testis, if you see, we have about 250 lobules, testicular lobules, and in each of these lobules we have 1 to 3 seminiferous tubules. Okay, now these seminiferous tubules inside have two kinds of cells. One is the spermatogonia, which is going to actually undergo the spermatogenesis and produce the mature sperms, whereas the other type of cells we have is the elongated cells known as the Sertoli cells. Now these are meant for nourishment and they participate in certain functions for converting the cells into the mature spermatozoa. Okay, I'll tell you in detail about it, but for now just remember that we have the two different kinds of cells present inside the seminiferous tubules. Okay, now once the sperms are ready inside the seminiferous tubules, we need a set of ducts. Okay, we need a set of accessory ducts to help us bring the sperm outside of the testis to the outside. Correct? So here what happens is, from the seminiferous tubule, okay, we have structures known as the retetestis, who will bring the sperms outside of the seminiferous tubule. Then this will connect to someone called as the vasa efferentia. This will bring it outside of the testis. Okay, now once from here we have the retetestis, then from here we have the vasa efferentia. On the outside we have a very supercoiled structure. How did it disappear? Okay, we have supercoiled structure which it's making fun of me. Okay, why is this disappearing? Anyway, so we have, never mind, I'm just going to draw it a little differently. Okay, so we have the seminiferous tubules, we have retetestis, we have vasa efferentia, and then we have a supercoiled posterior structure. This is definitely making fun of me now. Okay, wait, one last time. Abba, okay, it didn't disappear. How is it disappearing? I don't know. Okay, now this app is making fun of me now. Okay, enough. Now this is the supercoiled structure here, which we call as the epididymis. Okay, so this epididymis is nothing but a posterior structure which is going to be supercoiled. It will store the sperms until the ejaculation. Clear, everybody? Now this, yes, on the posterior side, Tarun Raghav. Okay, so this particular epididymis will store the sperms until it is going to be ejaculated outside. Now from this epididymis, we have one large tube, one long tube. Okay, after the epididymis, we have one long tube called the vas deferens. So this vas deferens is going to carry the sperms up into the abdomen. Okay, where into the abdomen? We have particular glands called as the seminal vesicles. Now this seminal vesicles will have a duct. This vas deferens will go and fuse with the duct of seminal vesicle and form what we call as an ejaculatory duct. And from the ejaculatory duct, it will be merged into the urethra, and from the urethra, it will pass through the external genitalia and it will open out into the urethral meatus. Meatus means opening. So it will open out through the urethral meatus. Okay. So if you look at the flow of the sperms from the retitestis, vasa efferentia, epididymis, vas deferens, then from the vas deferens it will fuse into the ejaculatory duct. From there it will merge into the urethra and open out through the urethral meatus. This is the flow of the sperm. They can ask you as a straightforward question. So remember that. Okay, is that clear, everybody? All right, okay. Now coming to the glands. Now it's not just enough for me to deliver the sperms outside. These sperms have to be alive and have to maintain its nourishment until it meets the egg for the fertilization, correct? So in this process, these sperms need to be nourished with secretions that come from three major glands, so which make up the accessory glands. Okay, so here we have firstly the seminal vesicle. Now the seminal vesicle is the one that contributes 70% of the semen or the seminal plasma. Okay, now here if you see, it contains a lot of fructose, okay, a lot of ions, okay, enzymes, all of this will be present inside this secretion. Then we have the prostate gland. Now the prostate gland contributes about 20 to 25% of the total seminal plasma, and this is going to give again some enzymes along with prostaglandins which can help the sperms to move inside the female's reproductive tract. Okay, now apart from that, last we have the bulbourethral glands. Now these bulbourethral glands secrete a mucus-like substance mainly meant for the lubrication during the insemination. Okay, and also it helps to neutralize the acidic pH that's found in the female's reproductive tract. Okay, is that clear, everybody? Beautiful. All right, so these are the three glands that are participating. So along with this entire secretions which we call as the seminal plasma plus the sperms together form what we call as the semen. Okay, now in this 40 to 60% of the sperm, so clearly 60% of the sperms should have normal shape and structure, okay, and at least 40% should have vigorous motility. Okay, is that clear to all of you? Now coming to the female reproductive system. Okay, here who is the main primary sex organ? The primary sex organ is nothing but the ovaries. Okay, we have two ovaries. Now in this ovary, we have a cortex where all the follicles are present. Okay, and then we have a medulla, which is that nourished with all the blood vessels and the nerves and all of that. Okay, so we have a medulla and we have a cortex, and it is all covered inside a germinal epithelium. Now from the ovary, we have tube-like structures that connect the ovary to the uterus. Okay, so here the funnel like structure in the beginning is called as the infundibulum. Okay, and this has this finger like structures called the fimbriae, which is going to pick up the egg as soon as it is released during ovulation. And then this infundibulum will lead to a slightly wide part called as the ampulla. Okay, this is where the fertilization takes place. So when the egg is coming here, at the same time, if we have a sperm that is coming to this place, only then it will lead to the pregnancy. Otherwise, it will not lead to any form of pregnancy. Is that clear, everybody? Is that clear to all of you? Beautiful. Okay, so from the ampulla, we have a short, narrow-like region called as the isthmus, which will connect the fallopian tube to the uterus. Okay, so this whole region is called as the oviduct, or also called as the fallopian tube. Now, this is leading into a inverted pear-like structure called as the uterus. Okay, now this uterus has 3 layers. The outermost layer is the perimetrium, which is fibrous and thick. Then we have a no, it is fibrous and thin actually. It's just a membrane at the end. Then we have a smooth muscle layer, which is the myometrium. Now, the myometrium is the one that undergoes serious contractions during the childbirth. And then innermost layer, we have the endometrium. So, this endometrium is the one that is very, very glandular in nature. It will secrete a lot of substances, and this is the one that undergoes periodic changes every month. Okay? Is that clear? Is that clear, everybody? Now, when you look at the uterus, the end of the uterus has a narrow neck-like structure called as the cervical, and in the between of the cervical, we have a short cervical canal. Okay, and this canal leads into a muscular tubular structure called as the vagina that will actually accept the sperms during insemination. So during childbirth, this cervical canal plus the vagina together make up what we call as the birth canal. Okay, is that clear, everyone? Okay, so guys, please pay attention to the class. Okay, let's discuss about everything else later. Okay, after your NEET exam, you can talk with whoever you want and do whatever you want. But now it's very important for you all to pay attention to the class. Okay, so we are running short in time. So let's focus only on the class. Is that clear, everyone? Is that clear, everybody? Okay. Perfect. All right. So now what we're going to do is we're going to move on to the external genitalia of the female. But any questions? Any questions, anybody? Okay. Right. So now let us focus on to our this thing. Okay. So I am going to now continue with the female external genitalia. Okay, now the external genitalia in the males is nothing but the penis. Okay, now the penis is a single structure with a lot of about 3 layers of erectile tissue that is present. Now these tissues help in erection of the penis in order to deposit the sperms into the female's reproductive tract. Now additionally, the end of the penis is a triangulated structure known as glans penis, and there is a layer of loose tissue, loose skin that is present around the glans penis, which is called as the foreskin or the prepuce. Okay, is that clear? Now when you look at the female's external genitalia, there is a mos pubis, which is nothing but a cushiony layer of tissue that is present along with pubic hair. And then we have 2 layers of folded tissue like structure, okay, which is called as the labia majora and the labia minora. Okay, now both of these together is going to contribute to the to the external genitalia. Is that clear, everybody? Joe Benin, Karippu, is it clear? Saran? Okay. All right. Cool. Okay. So after that, we have the clitoris, which is the sensitive tissue of the female external genitalia, and then we have the hymen, which is a partial membranous covering around the vagina. Okay, now this was considered to be an indicator of virginity, but later we found out that this hymen can break because of strenuous activity, stress, or any other reasons also, and not necessarily indicate the virginity for the female. Is that clear, everyone? Okay, is that clear? So that's all about the female and the male anatomy. Now we are going into the process of gametogenesis. Now remember that in gametogenesis, we clearly have to understand the process of cell division. First, we have 2n number of chromosomes. They are going to split into n cells at the end of meiosis 1, and each of these cells are further going to divide in order to form 4 cells at the end of meiosis 2. Clear? This is the basic cell division that we see in the case of meiosis. Now when you look at gametogenesis, the first thing we're going to look at is spermatogenesis. Okay, so spermatogenesis is where the sperms are produced in the male body. So we're going to have a starting cell called as the spermatogonia. Now this spermatogonia is 2n number of cells. Now this is just going to get up, get ready and prepare to go for meiosis, and this will be called as the primary spermatocyte. Okay, which is again 2n number of chromosomes. I have not started meiosis yet. I just got up and got ready, right? So now this primary spermatocyte is going to undergo meiosis 1 and produce 2 cells, which is going to be called as the secondary spermatocyte. Now remember that a male is always 44 plus XY. So at this stage itself, we see that the secondary spermatocyte would have become 22 plus X in half the number of cells and 22 plus Y in the remaining. Is that clear? Karpa, I'll answer your questions, Karpa. Just let me finish. So by the time I finish, you will have the answers with you already. Don't worry. Okay, so now if you see, the secondary spermatocyte will further undergo the meiosis 2 process, and it will give rise to 4 cells, which is haploid in nature, and these are called as the spermatids. Okay, so 4 equal haploid spermatids are formed. Okay, now this particular spermatid, okay, will mature into a proper spermatozoa. Okay, this process is known as spermiogenesis. And the Sertoli cells need to definitely help in order for the spermatids to convert into a mature spermatozoa. Is that clear for you? Yes, I will, Tarun. I'll teach you it even easier. Don't worry. So I will explain to you once I'm done with oogenesis also. I'll show you both. Okay. Alright. So now once the spermatozoa is ready, it has to be released out from the seminiferous tubules. Okay, so released from the seminiferous tubules is what we call as spermiation. Okay, Now is it clear to you, Karpe? So the process of converting a spermatid into a spermatozoa is spermiogenesis, and spermatozoa being released out from the seminiferous tubules is what we call as spermiation. Okay? Clear, everyone? So this whole process is what we term as spermatogenesis. Now this spermatogenesis starts only at puberty for the males, and it can go on lifelong, and it will only start during puberty. Remember that, because in oogenesis it's going to be slightly different. But before we go to oogenesis, let me tell you that the mature structure of spermatozoa will have a cap-like structure on the top known as an acrosome. Okay, and then there is a nucleus present in the head region. Okay, then after that there is a small middle piece followed by there's a small neck followed by a middle piece that has a spiral mitochondria. Okay, now this spiral mitochondria is called as a nebuncon. Okay, now this structure will end with a tail. So there is a head, neck, middle piece and a tail that is present in the mature spermatozoa. Okay, is that clear to you, everyone? Okay, that's a good trick, Tarun. Good job. Okay, perfect. Now when you look at oogenesis, what's going to happen is, try to understand clearly. It's going to be the same thing. I'm going to have 2N. Instead of spermatogonia, I'm going to have a oogonia here. Okay. Now from the oogonia, like how it differentiated into a primary spermatocyte, I'm also going to have a primary oocyte. Everything is the same, which is also 2N. This will undergo meiosis 1 and it will produce secondary oocyte, which is N number in nature. Now this will undergo meiosis 2 and it will form the ovum. It's all the same. But how it is different is, firstly, it will start in embryonic stage itself. Okay. So in the embryonic stage itself, the primary oocytes are produced, and once it enters the meiosis 1, it is arrested at prophase 1 and it will wait there. It will wait until this child comes to puberty. Okay. Now there are no other new primary oocytes that will be created after puberty. So strictly this particular cell is going to have a clear cut, maybe let's say 60,000 to 80,000 oocytes per ovary when it reaches the puberty. Okay, now from there what's going to happen is, it will finish the meiosis 1 as soon as puberty comes. The hormones will start working and then the secondary oocytes will be produced. Now again, it will get arrested at metaphase 2. Okay, so it will get arrested at metaphase 2. And at metaphase 2, it will wait until the fertilization is taking place. Okay, so when the fertilization is happening and the sperm enters the egg is when it will complete the meiosis 2 and a final ovum will be produced. This is the first difference that you have to understand. Now, Tarun was asking me, ma'am, show me with the diagram that we see in NCERT, which is what I'm going to show you now to explain how different spermatogenesis is from oogenesis. So firstly, we have one spermatogonia. Okay, this is going to differentiate into one primary spermatocyte, which is also 2N. Okay, and this will divide into two secondary spermatocytes, which is N number, and then further they will give rise to 4 haploid spermatids. Correct? And this will now mature to form the final spermatozoa and my spermatogenesis is complete. Okay. Now if you look at oogenesis, same thing. It starts with 2N. Then I have a primary oocyte, which is 2N number of chromosomes. Now here itself, this is embryonic. Okay. Now is the point. When this is dividing into a secondary oocyte, it will not be equal like it is in the case of spermatocyte. Instead, what will happen is one small N polar body is released and the other one is forming a secondary oocyte. Only one single secondary oocyte is formed, that is N number of chromosomes. Okay, now again this is going to arrest, okay, before it forms the ovum, and once the fertilization starts and the sperm head is entering the egg, it will complete the meiosis 2. Again, all the cytoplasm is released in the one major ovum, one functional ovum, and it gives rise to the second polar body. Okay, is that clear to all of you? Yes, Tarun, just before the fertilization. So fertilization is nothing but fusion of the nucleus, right? Just before the fusion, once the sperm head will enter the egg, it will complete the second meiosis. Okay, clear to all of you? Perfect. Okay. Tarun, Raghav, yes, acrosomal enzymes contain hyaluronidase, but you don't have to know too much details of it. It's not required for your exam. Okay? That's it. Okay. So here, sometimes they will say 3 polar bodies. So that time you should not get confused. They're just trying to say that this for the polar body, it can also form 2 others. Okay, which will also end up degenerating. So end of the day, out of 4, only 1 is functional and 3 will degenerate, just like in the case of megasporogenesis that we saw. Clear to all of you? That's it about your gametogenesis. Okay, any doubts in gametogenesis? Okay, so Karpa, the function of polar bodies is not required, Kanna. So just remember that the polar bodies are actually just the namesake chromosomal element. Okay, so when you're dividing 2 N chromosomes, one has to be N, the other has to be N. All the cytoplasm is retained by only one cell, and something so small is thrown out on the other, because we need to maintain the zygote until it is getting implanted. So who will give the nourishment? So completely it will be the cytoplasm, the elements in the cell that will support the zygote until it will go and attach to the mother's body. Clear, everyone? Okay. Perfect. Okay. So now once we have done with the gametogenesis process, the insemination will take place, and if at all the sperm and the egg are coming to the same place at the same time, then the fertilization will happen. Now we have to understand menstrual cycle. Okay, what exactly menstrual cycle does? What is this cycle? It is the periodic changes that undergo in the female's body. Okay, so before that I'll tell you the hormonal regulation also. Okay, so then I'll go into the menstrual cycle. So firstly, in the males, what happens is the GnRH will be released from the hypothalamus once the puberty is attained. This GnRH will ask the pituitary to release 2 hormones, which is LH and FSH. Now, LH in the males will act on the Leydig cells, the interstitial cells, okay? So, this is also called as the interstitial cell stimulating hormone, okay? Now, this Leydig cells will contribute to the actual spermatogenesis process, all giving rise to the male secondary sexual characteristics. Everything will be taken care of. Now, this FSH will be somebody who will act on the Sertoli cells, who will release certain factors that are essential for the conversion. Remember, I told you spermiogenesis. So for that, this FSH is super useful. Okay, understood? Now, in the case of the females, how the hormonal regulation works is, once the GnRH has released during puberty, it will again ask it to release 2 hormones, LH and FSH. Now this FSH will go and increase the follicles, will go and stimulate the follicles to grow, and these follicles will release a hormone known as estrogen. Okay, now this LH will actually have a surge in between the cycle, which will contribute to ovulation and further developments in the ovary to happen. Okay, now if you look at the entire process. Okay, so firstly, menstrual cycle is, like I said, the periodic changes that happens in a female's body, and this is basically happening at intervals of about 28 to 30 days. Okay, is that clear, everybody? So this particular menstrual cycle starts around the age of puberty. Okay, now why this happens during the time of puberty? The first menstruation we call as menarche. Okay, and why it happens only around puberty? That is when the GnRH will be released. Okay, is that clear to all of you? Right, wonderful. So now this menarche is the start of menstruation in the woman, and menopause is the end. Okay, is the end of menstrual cycle. So unlike the males, it will not happen throughout life for the women. Okay, is that clear to all of you? Now, coming to how and what exactly happens during this menstrual cycle. So, firstly, what happens is, I'll show you both the changes in the ovaries as well as the uterus. Okay. So, first phase of the menstrual cycle is the menstruation phase. So, in the menstruation phase, what is happening? The endometrium will break down. Okay, it will break down, and then bleeding will be there for about 3 to 5 days, and all the tissues and everything will be chucked out. Okay, and in the ovary, my structures have degenerated and a new follicle will be selected for growing further. I'll come back to this phase. Now, the second phase that we have is called as the follicular phase, or also called as the proliferative phase. So here what is happening is, in my ovary, one new follicle is selected, and this will start growing the follicular cells. Now these follicular cells will secrete estrogen, which will go to the uterus and ask it to start dividing. Okay, so proliferation is happening in the uterus and the follicular cells are growing in the case of the ovary. Okay, now this primary follicle becomes a secondary follicle, and then slowly it will develop some water pockets inside. Okay, and this is what we will call as a tertiary follicle. Now this will mature into a proper Graafian follicle. Okay, so all this follicular growth is taking place in the follicular phase. Okay, now this particular Graafian follicle will have the mature secondary oocyte that is present inside. Now what happens? Now this phase is about 9 days long. Okay, now after that, the third phase is the ovulatory phase, wherein the LH surge will happen, the Graafian follicle will rupture, okay, and it will release the egg. Ovulation is going to take place. Okay, happens around the 14th day, or we can say middle of the cycle. Clear, everybody? Now after this, the last phase is the secretory phase or the luteal phase. Why is that? Now once the Graafian follicle has released, the egg has gone, but the remaining follicular cells will now convert to a structure known as a, again it disappeared. Okay, again. So it will now divide and form a structure. Okay, it will form a yellowish structure called as the corpus luteum. Okay, now this particular corpus luteum is going to produce a hormone called the progesterone. This progesterone will go to the layers of my uterus and proliferate it more and more and more and more and more. This app is making fun of me. Okay, now again it will make the uterus to develop more and okay, you all understood? No, so the uterus is proliferating. Okay, it won't disappear off like this, but anyway, yes, so it will proliferate, Shaba, and it won't disappear. Okay, so it will just proliferate and it will become really glandular in nature in order to accommodate the baby. Okay, I don't know why this app is making fun of me today. Okay, Janda, stay there. Okay, stay. Okay, so now if you see this particular stage will enhance the growth and proliferation of the endometrium, and it will make it extremely glandular and secretory for all the nutrients to circulate, so that when the implantation takes place, the baby can be taken good care of. Okay, now this is the time when all the body is functioning expecting a pregnancy. Now this will wait for about 14 days. Okay, if there is no signal that is coming from the newly developed fetus, if there is no pregnancy, then what happens is it will directly cause the degeneration. Okay, so it will degenerate about around the 25th or the 26th day of the cycle, and what will happen is it will go back to square one, where the endometrium will break down and then all my tissue will get out. Whatever I had developed, everything is gone in the absence of fertilization. Clear, everybody? Is that clear to all of you? Yes, good question, Tarun. So that is if pregnancy does not happen. If pregnancy happens, what happens is as soon as the zygote is formed, okay, what happens before I go to that, I'll talk to you about fertilization. Okay, so fertilization is nothing but the process of fusion of the male and the female gametes, right? So this is my egg. The egg naturally has a layer known as a zona pellucida. Okay, now this zona pellucida will be covered by another spindle-like layer of cells which we call as the corona radiata. Okay, now what happens is when the sperm is coming, okay, it will penetrate through the corona radiata and reach the zona pellucida. As soon as one sperm enters the zona pellucida, the entire zona pellucida will begin to thicken, its walls will begin to change the polarization of its wall so as to prevent any further sperms from entering the egg. Is that clear, everybody? Yes, definitely, Kundubagar, no problem. Yes, Tarun Raghav, it's a non-cellular layer. Okay, so zona pellucida is a non-cellular layer. Okay, so the sperm will enter the zona pellucida, fuse with the plasma membrane, and once it has entered is when meiosis 2 is complete, and then the fusion will take place, which we call as syngamy. Okay, is that clear, everybody? Yes, absolutely, to prevent polyspermy. Good, Tarun. Okay, now once this is done, this particular zygote will have one nucleus and this much amount of cytoplasm. The same cell will not grow, but it will form 2, 4, 8 and a 16 cell stage. That is when we will call the cell as a morula. Okay, so what we see here is that the morula is nothing but a ball of cell. Again, morula disappeared. Okay, let me make it a little more cleaner. So the morula is going to have a bunch of about 8 to 16 cells in it, and all of these 16 cells, I think the app is telling me, ma'am, you need to draw diagrams better, but okay, I'm only drawing it for your understanding, so hope you understand. Anyway, all these 16 cells are going to be identical. They don't have any differences in them. Okay, so these particular cells will now undergo a differentiation when they are becoming 16 to 32 cell stage. So from a morula, it is becoming a blastocyst. So here I'll have an outer trophoblast layer, okay, and an inner cell mass layer. Okay, so the inner cell mass is the actual group of cells. Again, inner cell mass disappeared. Okay, bro. Okay, app, don't worry. I'll try to make my diagrams better from next class. Okay, don't torture me like this. Anyway, so, okay, I'm just going to draw cells. I think that's what the app also wants me to do. But anyway, there is a inner cell mass. Okay, so this inner cell mass is going to develop into the actual embryo. Understood, everybody? So what's going to happen is these trophoblast layers are going to develop some extra embryonic structures around the embryo. Okay. Yes, Joe Benin, thank you so much. Pakka. Okay. Right. So what happens is, in the uterus, when this blastula has come, so by the time, it is getting fertilized, it will become 1, 2, 4, 8, 16 cell, 32 cell stage. It's already come here. Okay. Now this will develop a structure known as the chorionic villi, and it will go and implant itself into the uterus. Okay, as soon as this implantation, which is nothing but attachment of the cell, is over, this is going to release something called as HCG, human chorionic gonadotropin, who will tell the corpus luteum not to go. He will say, bro, I have come, please don't degenerate. And the corpus luteum continues to give progesterone, continues to give as much hormones as possible to maintain and support the pregnancy. Okay, is that clear to all of you, everyone? No, Tarun, it's not HPL. I'll tell you where HPL will come. Okay, here it is HCG. HCG will tell the corpus luteum to maintain. Now, once the implantation is complete, we will go into the actual process of gestation, which is nothing but the pregnancy. Understood? Yes, Arun. We are already done with more than 10 to 15 chapters, I think. So we just have another few more, which we will finish in another couple of days. Okay. Pakka. So now this particular gestation process, okay, is how this inner cell mass and all of these cells will develop and become a whole baby. Okay. So here the inner cell mass will now undergo differentiation of layers. Okay. So it will form an ectoderm, endoderm and a mesoderm. And from here, one organs will start forming. So this stage is called as a gastrula. Okay. Now from here, by the end of the first month, we'll have a heart. Okay. By the end of second month, all the limbs, digits, everything has been formed. And then by the end of the third month, which is the end of the first trimester, what happens is all the vital systems are done. Okay, by the end of 5th month, you can see movement. Okay, and by the end of 6th month, which is the end of the second trimester, you can see hair, eyelids, okay, eyelids and all that differentiated. And then last, in the third trimester, it is only growth and development of the baby. Okay, now when the baby is getting implanted, there is some tissues from the fetus and some tissues from the maternal side also together, which builds a new organ altogether, which is called as a placenta. Okay, so this particular placenta is going to produce HCG going to produce HPL, which is human placental lactogen, like who told me? I think Tarun asked me, no, where HPL will come. So that is where HPL will come. It will help in differentiating the mammary glands to produce milk for the nourishment of the baby. Apart from that, a lot of progesterone, a lot of estrogen, all this is produce. Okay, now this particular placenta is the connection between the mother and the child, so it will give all the nutrients, antibodies and anything needed for the baby, and the baby will also, you know, give out all the carbon dioxide, the waste materials and everything back into the placenta itself. So that is basically the exchange platform for the baby and the mother. Okay, now apart from that, in the mother's body you will have cortisols, okay, then thyroid hormones, okay, and then again progesterones, estrogens and all of these hormones which are multiple folds. And why should there be so much of hormones in the body? Because it has to support the making of one whole organism on itself. Okay, so that is why it is all there. Now once the baby has fully developed, the placenta plus the fetus will send out a fetal ejection reflex, okay, which is nothing but mild contractions. Okay, some small contractions it will release to tell that, okay mom, I'm ready. Now looking at this reflex, the pituitary of the mother will start releasing oxytocin, and this oxytocin will make stronger contractions. Okay, now the contractions will again tell the oxytocin, then oxytocin will make more contractions. Now this will repeat until the baby is pushed out. Okay, and this entire process is known as parturition, which is the birth of the baby. That's all. Okay, and once the birth of the baby is done, the same oxytocin, once the birth is complete, will act as a milk ejection hormone, okay, and will allow the milk to flow out of the mother's body. Is that clear? Now this particular milk that is ejected, especially the first milk, is called as the colostrum. It has extremely high amounts of IgA, and that is very, very important for the immunity of the baby. Okay, is that clear everybody? Yes, Arun, I have already discussed all the chapters. Okay, today we'll be finishing all the 12th and we're already done with half of 11th also. So I'll be doing separate sessions for discussing the questions, don't worry. Yes, Tarun, it is positive feedbacks because more contraction, more hormone, again more contraction, again more hormone. So that is why it is called as a positive feedback mechanism. Okay, clear everyone? Now for the pathway of the milk, remember when I taught you the reproductive system, I didn't teach you mammary glands. So let me just revise that for you, that the milk flows from the cells, secretory cells in the mammary glands called as alveoli. Now from the alveoli, okay, we have a set of mammary tubules. Okay, now from the mammary tubules, we have mammary ducts. From the mammary ducts, it will open out into the mammary ampullae. Okay, from the mammary ampulla, it will open out into the lactiferous ducts, and from the lactiferous ducts, it will open out into the nipples from which the baby will be suckling out the milk. Is that clear? Clear everybody? Pakka. That's it for human reproduction chapter. Any other questions, anybody? Any other questions, anyone? Anything that you all want me to discuss or any doubts that you all have? Okay, Karp is asking for trick. See, how I used to remember is from the alveoli, we have tubules, right? Tubules, ducts, ampulla, duct. So now the next duct after the ampulla will not be a mammary duct again. It will be a lactiferous duct. So like that, you can just remember tubules to duct, duct to ampulla, ampulla to duct, and then duct out into the nipples. Okay. So yes, like Tarun is saying, we cannot have mnemonics for every single thing. Then you will have a separate master bit to remember all the other mnemonics also separately, which is difficult. Okay, so we will not go into that. Okay, but I hope this entire thing is clear to all of you, everyone. Yes, everybody? Perfect. Okay. Now coming to reproductive health, guys, I'm going to keep reproductive health extremely simple. Within 10 minutes, we can learn entire reproductive health. Okay, it's not a difficult chapter at all. If you see, when you look at reproductive health, the first introductory part talks about the different population explosion that has happened, and importantly, the reproductive and child health care programs. Okay, what it has done. So simple general knowledge. Okay, so if you want a reproductively healthy society, everybody should be aware. They should know how to take care of the mother when she's pregnant, the mother after she delivers. You should know how to take care of the baby. You should know why you should vaccinate a child. So these kind of things are very important to educate to the people. Secondly, making them understand about population explosion. Okay, making them understand about the benefits of having a smaller family is very important. Because let's say a certain family is earning so much and they have 10 children, then imagine they'll have to provide and they have to give for all the 10 children, and as a result of which the standard of their living will never improve, right? But if they have maybe 2 or maximum 3 children, let's say instead of 10, so then what happens, a smaller family can definitely build more resources to provide for the children. And also as a country, if we have limited population and a good number of individuals who can contribute back to the society, then what happens? We are just at a benefit of maintaining and utilizing the resources to the maximum. Okay? Is that clear, all of you? So that is the first part in the RCH programs. I'm not going to the details of it. Now the most important part of this chapter is contraceptives. Okay? Now when you look at contraceptives, okay, I'll tell you the contraceptives in the different methods that are involved. So contraception is nothing but preventing an unwanted pregnancy. Okay, so not all intercourse should lead to a pregnancy. So in order to prevent any unwanted pregnancy, the kind of methods and chemicals that you can use for it come under the contraceptives. So firstly, we have the natural methods. Okay, so natural methods are very effective in terms of not having any side effects, but unfortunately the rate of failure is very high. Okay, so firstly we have the periodic abstinence. So periodic abstinence is where there is no intercourse between 10 to 17 days of the menstrual cycle, because that is the expected fertile window where the ovulation might take place. So if there is no intercourse during that period, then that means there is no chance of getting pregnant also. Now the second one is coitus interruptus. Okay, this is where the male partner will withdraw the penis before the ejaculation. So if the sperms are not deposited, again there is no chance of pregnancy. The third one is lactational amenorrhea. This is where soon after the childbirth, the women may not be having her menstrual cycles because the brain is not releasing any more GnRH. Okay, so this is only effective to a period of 6 to 8 months after the delivery, where the mother will not have any ovulation because she is intensely breastfeeding the child. Okay, but this is not effective after 6 months. So these three methods are natural methods where you are preventing the chance of meeting the egg of the sperm meeting the egg at all. Now the other one, what happened? So the other method is barrier method. Okay, so in barrier method, what is happening? In barrier method, we are creating a barrier. So that includes condoms, diaphragms, okay, vaults. So this is meant to prevent the sperm from entering the reproductive tract. So that means there is no physical meeting, and as the result of which there is no possibility of pregnancy again. Okay, now this is the barrier methods. Now after barrier methods, we have the IUDs, which is nothing but the intrauterine devices. Now remember, examples of intrauterine devices is super important. So firstly, we have the non-medicated IUDs. Okay, examples are lipase loop, please remember. Okay, then the other one, so non-medicated means they will just improve the phagocytosis of the sperms, and that's about it. Then we have the copper releasing. Okay, so in copper releasing we have CU7, CUT or multilode 375, all of which will release copper. It will interfere with the fertilizing capacity of the sperm and also with the motility of the sperm. So the sperm cannot climb up and fertilize the egg. Okay. Now the last one is the hormonal IUD. So hormonal IUDs will continue to release some hormones. Example, LNG20 or progestasert. Okay, so all of these will release hormones in a way that even if fertilization happens, the uterus is not suitable, the endometrium is not suitable for implantation. Is that clear? Now, apart from IUDs, one can go for oral pills. Okay, these are nothing but plain progestogens or combination pills. So they will entirely inhibit the ovulation. So it will tell the body that, bro, you finished ovulation, da, see, progesterone has come, and the body also will be fooled and it will think, oh, okay, is that the case? And it will prevent the ovulation entirely. So there is no chance of getting pregnant. Now, instead of taking these hormones as pills, you can also take them as injectables or implants, okay, wherein it will be there in the body and it will give a longer effective period. Okay, now in the case of a unprotected sex at some point and for emergency contraception, either one can go for emergency pills or take an IUD immediately so that the pregnancy can be avoided. Okay, now the last technique of contraception is the surgical procedures. Okay, so in females it is called as the tubectomy, in males it is called as the vasectomy. Okay, so in females we're going to cut and stitch the fallopian tubes. So even if ovulation happens, the egg cannot cross and come to the uterus, and the sperm also cannot reach the egg. So no so there will be no possible fertilization at all. Now, vasectomy is the case where we will cut the vas deferens and we will stitch it. So, as a result of which the sperm will not be deposited into the female tract at all. Okay, so the sperms will just degenerate and it will die there only. It will not come to the outside. So, as a result of which you can understand that there will be no chances of pregnancy. Both of these are sterilization techniques and these cannot be reversed. Okay, it is an irreversible technique. Some people who take this very early in their life can probably, have a reversibility, but that is also very, very poor. Okay, but usually the people are sterilized. Okay, so it is an irreversible process. Is that clear to all of you? Now coming to the next part, which is sexually transmitted diseases. Any disease that can spread through an intercourse is what we call a sexually transmitted disease. Now remember 3H, okay? HIV, herpes, genital herpes and hepatitis B. Now these three diseases have no cure. Apart from that, we have bacterial and viral diseases like gonorrhea, chlamydia, okay, trichomoniasis and so many more. All of these can be cured if you find it out at the earliest stage. Is that clear? Now, all of these are majorly spread because of, having multiple sex partners, having unprotected sex, or, because of blood transfusions in the case of HIV or hepatitis B. And in the case of babies also, it can transfer from the mother to the fetus in the pregnancy itself, okay, through the placenta itself. So these are the chances of getting the disease. But how can you prevent yourself from the disease? Obviously, you should avoid having intercourse with unknown and multiple people. And again, it should be taken care that when you're taking a blood transfusion, the blood banks should run checks for HIV and other diseases so that it does not spread to the patients, okay. And also before pregnancies, they do HIV tests so that the child can be saved from it. Now, because HIV, hepatitis B and herpes has no cure, you can only give some drugs to prolong the disease, but you can never cure them. They will die at some point. Okay? Is that clear to all of you? Yes. Now coming to the last part of the chapter, which is the artificial reproductive technologies. Now infertility is a thing wherein a couple has been trying for 2 years but they are not able to have a child, then it will be called as infertility. Okay, now if you look at the artificial reproductive technologies, these are ways you can help the such couples to have a child. How do we do that? So if you see in a test tube baby program, okay, so what is the test tube baby program? We are going to fuse the gametes in the lab, which is called as in vitro fertilization. So outside the body we're going to fuse the cells, okay, and we're going to transfer the embryo transfer. So either we can transfer it directly in the fallopian tube, like just after the fertilization, which will be called as it will be called as zygote intrafallopian transfer, which is ZIFT, or we can transfer it directly into the uterus after it comes to about 16 to 32 cells, which we call as a intrauterine transfer. In both these cases, I will fuse it outside the body and put it back inside the body once the mother is ready. Got it? Now, in case of the female not able to produce the gamete, we can do a gamete intrafallopian transfer where we'll take the eggs from a donor and we'll put it inside the woman. Then as usual, fertilization can happen and continue with the pregnancy. That is also possible. Now, in cases where the male is not able to inseminate, we can do artificial insemination, or we can also do an intrauterine insemination directly in order to bring about the insemination or to deliver the sperms into the female's body. Okay. Now another important technique is called as intracytoplasmic sperm injection, which is nothing but directly injecting the sperm nucleus into the egg and hoping that the fertilization will be carried out. Okay, so these are the different techniques that you can utilize in order for the couples to have a child if they are not able to conceive naturally. Clear, everybody? That is it. We are done with the entire reproduction unit. Those of you who have not subscribed, subscribe the channel and do join the Telegram channel for all the free notes. And now we are going to continue on into our next unit, which is human welfare. In that we are going to do human health and disease. Are you all ready, everybody? Yes, are you all ready? Can I see some energy in the chat box? Throw some fire, throw some stars, and let's do human health and disease, okay? So when you look at human health and disease, okay, it's a very, very simple chapter and I'm going to teach you in a way that you will remember it very easily, okay? So listen to my explanation, because why I say it is, now if you can just some people ask me, ma'am, can I just directly see the slides once you send it in the Telegram group? But that will not work, because most of the things I don't write, I will be explaining here. Okay, which is what I want you all to analyze and understand, so that you don't make a mistake in your exam. Okay, clear, everybody? So let us start off. So when you look at human health and disease, okay, fundamentally what you will see is, in human health and disease, you will have to study the diseases. You have to study what immunity is, what your immune system does for you. You need to learn the two diseases, AIDS and cancer. You will have to have a small note about the drugs. This is the most important parts of this particular chapter. Okay. Now, starting with the disease, we have bacterial disease, which is typhoid and pneumonia. Okay. So, typhoid is caused by Salmonella typhi. And pneumonia is caused by two species. One is Streptococcus pneumoniae and the other one is Haemophilus influenzae. Now remember, most probably they will not ask you Streptococcus pneumoniae, because pneumoniae, they know you will find the answer. Okay, so 100% they will ask you questions like Haemophilus influenza, because that is the one that is confusing. So remember, it is going to cause pneumonia. Okay, now Salmonella typhi is caused by contaminated food and water. Okay, so because of that, you can say that, you know, that it will have problems in the stomach. In very worst cases, perforation of the intestines can happen. It can cause death also. But overall, you will see that contaminated food and water is the major reason why you get this disease, and you can find this disease by a Vidal test to diagnose the disease. Okay, now if you look at pneumonia, it is majorly because of the aerosols. It is a respiratory disease. So when someone else is sneezing and you are standing next to them, you can easily get this disease, or by sharing objects with them. So the lungs will be affected, and there will be fluid that is filled in the lungs. So if there is fluid that is sitting inside your lungs, you cannot exchange the gases effectively, as a result of which you will have breathing problems, and it is a very important disease to monitor. Okay, so these two diseases are bacterial diseases. Okay, now next we will look into the viral disease, is common cold. Okay, and this is caused by a rhinovirus. I'm pretty sure nobody is there who has not gotten a common cold. So this virus sits inside, it mutates, it will cause a lot of running nose, sore throat, mild fever, headache, sinus issues, and all of these things. But it only affects the upper respiratory tract. It will not go into the lungs. Okay, so all your common cold is not pneumonia. Remember that. Okay, now after that, coming to the fungal diseases. So fungi, mainly the disease you need to know is ringworm. Okay, so ringworm is a disease that's caused by epidermophyton, okay, microsporum, or trichoderma. Okay, trichophyton. So these different species will cause the fungal infection called ringworm, where you will have an itchy skin, okay, itchy skin, scalp, nails. It can happen anywhere, and it can spread through using combs or clothes or even towels between the patients. Okay, so it can spread easily. Oh, I'm so sorry. Did I write it as trichoderma? No. Trichoderma comes in your this thing. Next chapter. Okay, human welfare in microbes in human welfare. But yes, trichophyton. I'm so sorry. Okay. Yes, so trichophyton. I miswrote it. Okay. Yes, so you will have lesions that are very, very itchy and dry, causing this particular disease. Okay. Apart from that, you have helminth diseases. So under helminth diseases, you majorly have ascariasis, which is caused by the Ascaris roundworm. Okay, and then you have the filariasis. Okay, we have the filariasis. Okay, and filariasis is caused by the Wuchereria, which is nothing but the filarial worm. Okay, Wuchereria bancrofti or Malai. Okay. So these two are going to cause. Now what this filariasis, what it does is, it will chuck the filarial worm into the body, and this worm will go and sit inside the lymph vessels, lymph nodes of the lower limbs majorly. So because they are sitting inside, now the lymph vessels will start to swell. So it will swell and sometimes cause very gross deformities in the body and in the genital organs as well. Okay. Now 1, 2, 3, 4, the last set of diseases you have to study is the protozoan diseases. So in the protozoan diseases, firstly you have amoebiasis. Okay, it's caused by Entamoeba histolytica. So this particular protozoan is spread through contaminated food and water. Okay, but majorly who is the carrier for this? It is the housefly. So the housefly will sit in the feces of somebody who is affected. It will carry the protozoan, come and sit on your plate of food also, and then nicely you will have that food, and then once it goes inside, all the protozoan will go inside and start showing its activities. Okay. Yes, Tarun. It is there in human welfare, the microbes in human welfare when we're talking about the biofertilizers. I tappa edified here. Okay, I wrote it wrongly. Yes, okay. Perfect. So here, when you look at the protozoans, which is the house flies, it's going to carry it directly onto your plate and you're going to eat it and get affected. Now, the other major disease you need to study is malaria. Okay, it's caused by Plasmodium, and this particular Plasmodium is going to take care of, it's going to have four different species that's associated with it, which is going to have Plasmodium malariae, then Plasmodium falciparum, okay, then Plasmodium ovale, and one more, Plasmodium vivax, okay. Now these four may say this is the one who is the most dangerous, okay. Is that clear? Okay. Yes, Tarun and Joe. Yeah. Okay, so coming to this, it is the most dangerous and sometimes can cause death also. Okay, so what happens here is typically this Plasmodium requires both mosquito as well as human hosts to complete its life cycle. Okay, so this particular Plasmodium will enter the humans, okay, in the most infectious stage called as the sporozoite. Okay, it will go to the liver, it will damage all the liver, it will undergo asexual reproduction, again it will burst the liver cells, it will go to the RBC, there it will utilize the hemoglobin and make something called as the hemozoin, okay, and it will burst the RBC also. Now in the process of bursting the RBC and releasing the hemozoin, you will start getting chills every evening or every 2 to 3 days, and that is the major symptom for malaria. Okay, now this particular RBC, I mean, as it is undergoing the asexual reproduction, it will release the gametophytes. Okay, but these gametophytes cannot fertilize inside the human. That is when it will go to the mosquito. When one mosquito will bite this person, this gametocytes will go into the mosquito. It will go fertilize in the gut of the mosquito, okay, and then it will go to the salivary gland and store as what I call as this sporozoite. Okay, so this is the mosquito, this is the human. So without both, this guy cannot survive. Understood? Yes, Mahesh. Okay, clear everyone? Is that clear to all of you? Perfect, and that's all about the diseases. Okay, coming to the immunity. First, remember that we have an immune system that is working for us. The primary lymphoid organs or the primary immune organs are nothing but the bone marrow, okay, our bone marrow and the thymus, okay. Now from here, it goes to the secondary lymphoid organs. Who are the secondary lymphoid organs? It's the spleen, then Peyer's patch, okay, then your tonsils, okay, mucosa associated lymphoid tissue and so on. All of these are going to be part of the secondary lymphoid organ where I get actual exposure to the antigen and it will fight for you. Okay, so this will make the immune cells, and here is where the actual immune cells will fight and protect you. Is that clear? Mahesh, it's all a PYQ. It's a previous year lookalike, Kanna. So all the questions in the AI tool are completely NEET oriented. You don't have to worry. Okay, so it's completely aligned with the previous year syllabus itself. All the previous year papers are loaded into it. Okay, all right. Okay, now when you look at immunity, it is the ability that the body is able to protect us from so many diseases that we are able to see. Okay, now in that, the first category is the innate immunity, which is there by birth. Okay, so under innate immunity, we have the physical barriers like the skin, who will not even who will not even allow any of the pathogens to come inside. Then after that, we have physiological, which is all the tears, the secretions, the acids and all that in the body that can kill. Beyond that, if it reaches our blood, then we have the cellular barriers, okay, who will fight for us and who will make sure that, who will kill any foreign item who comes inside, which is basically all the natural killer cells, all the various kinds of cells of the immune system who will basically fight irrespective of who you are. Okay, and then the last one we have is the interferons. Okay, so these are the chemicals synthesized by the affected cells to protect the other cells. Okay, now the other category is the acquired immunity. Now if you look at acquired immunity, naturally you can get this immunity by actual exposure to the antigen. Okay, so once you get exposure to the antigen, you develop antibodies to fight, and that will be very, very specific for each and every disease that is affecting you. Okay, now in this we have active immunity and passive immunity. Okay, so here making of antibody is involved. Okay, but here it is ready-made antibodies. Okay, so here what happens in making the antibody? Either naturally through exposure, you get the disease, you go flat for 7 days, you get full viral fever or you get full disease, you are affected, and then slowly you will recover and then build your antibodies and you will generate the antibodies on your own. Okay, so the next time you see the same pathogen, you can perform better. Okay, but the other thing is through vaccination. Okay, where we are introducing a dummy fellow inside and we are creating memory for these cells so that they'll be able to act better. So always the primary response is slow. Okay, secondary response is intense. It's very, very intense. Okay, so in order to get the secondary response, I'm going to vaccinate the person so that actually when I'll see the disease, it will not think twice. It will come, fight and done. Okay, so that will be the case for the secondary response, and this we can use because of natural exposure. First time you get the disease, second time you fight, or first time you get vaccinated and second time you fight. Okay, now when you look at passive immunity, now if a snake bit you, you need to understand that the snake bit you, then you have to start making antibodies and then fight that antibody, you will die by that time. So in order to save yourself from this kind of venoms or tetanus and all that, directly you can inject the antibodies from a horse or a rabbit where already this will be prepared and kept, and you can just inject the serum inside and protect yourself. Okay, now the other type of passive immunity is IgA through the colostrum, right? Naturally the mother is giving the ready-made antibodies to the baby, so all of these will be considered as passive immunity. Now this is one time, okay, and short-lived. If you get bitten by a snake one more time, you have to again go through this whole process and you will have to take the antivenom or you have to take the antitetanus. Okay, so for snake bites, for tetanus kind of diseases, you will always go for a passive immunity. Okay, is that clear to all of you? That's it guys. So we are done with immunity as well. Now we are going to this disease called HIV. So HIV is nothing but human immunodeficiency virus, okay, and it causes a disease called as AIDS, which is an acquired immunodeficiency syndrome. So it is acquired, it cannot be genetic, it cannot be there when you are born, it is something you get because of exposure in your lifetime. Is that clear? Now this particular disease majorly spreads because of intercourse, okay, because of blood transfusions, because of sharing needles during drug abuse, okay, and different and from the mother to the fetus also if the mother is affected. Okay, so these are the popular causes of getting the AIDS. Now is there prevention? Definitely there. So you can prevent AIDS by, avoiding sex with multiple partners, with unknown partners, checking the blood before transfusion, and also not sharing the needles in the case of drug abuse and other reasons. Okay, so obviously the syringes have to be very, very clean and maintained sterile. Otherwise, it's very easy to get this disease. Okay, now the mechanism of the disease is the most important thing you have to understand here. So what happens in the mechanism is this particular HIV has a DNA, a single strand. It has actually an RNA along with a reverse transcriptase enzyme. Okay, so what this reverse transcriptase is going to do is going to produce a double-stranded DNA, and then there is an enzyme called integrase which will help it to attach into our genome. Okay, so majorly this guy will go and sit in the macrophages, who is basically called as the factories of HIV. Okay, this guy will go and tell, bro, I am your DNA only, bro. I'm just here. Believe me, okay? I'm not a snake. I'm not going to betray you. Come on, I am your brother. I'm your sister. I'm your friend, da macha, like that. It will come and integrate inside the host, and this macrophage, poor fellow, will give place, and the HIV will start multiplying multifold, and it will make new HIV particles from there. Okay, now these guys will go to the T helper cells. Okay, majorly the T helper cells, and these T helper cells are the ones that help T cells and B cells to fight. Now, once these cells are attacked, my entire immune system will crash. That's all. So then any very bacha type ka infection comes also, you will get severely affected and you can die. Yes, absolutely, Tarun. Okay. So what happens is, no, Tarun, don't worry. They will not ask those kind of questions and all. Okay, they are very, very sure to ask questions from the NCERT itself. So don't worry, they won't ask you such questions. Okay, clear to everyone? All right, perfect. So this is the mechanism by which this HIV will react. Now, the other thing you should know is, how can I diagnose it? Okay, I can diagnose it through ELISA, okay, which is a very, very efficient technique. Even if very, very small micro units of HIV is there, I can easily find out and I can fight it. Okay, now the other thing is, apart from ELISA, the treatment. Okay, apart from the diagnosis, if you look at the treatment, AIDS has no cure. But I can give some antiretroviral drugs, I can prolong the disease, I can save this fellow from dying very soon, but death is inevitable. I cannot cure the disease completely. That is not possible at all. Okay, is that clear? So Tarun, the diagram is there in your NCERT, but see, they won't ask you to draw diagrams anyway in your NEET exam, and don't break your head over very micro details of the diagram. I'm explaining the mechanism to you, so just understand the mechanism and that will be enough for you. Okay. Is that clear? Now, the other disease that you should know and study about is cancer. Okay, globally, this is one of the major causes of deaths in the last 1 decade. Okay, and we have multiple researches that's happening around cancer and around cancer patients as well. Okay, now when you look at cancer, majorly what is happening, one cell is there. This cell is undergoing division. Okay, after this division is over, he has something called contact inhibition, where the guy next to him will tell him, okay bro, hi, nice meeting you. So this guy will be like, oh hi, okay, I met this guy, so okay, I am done with my replication or my division. But because this contact inhibition is gone, I don't care who is sitting next to me, and I forgot, did I finish dividing? And I'll be like, okay, let me divide one more time. Did I finish dividing? Okay, let me divide one more time. And there will be uncontrollable cell division. Is that clear to all of you? So uncontrolled cell division will be there. Lack of contact inhibition will be there. So he does not have eyes to see who is next to him. Did I finish or did I not? And as a result of which, I will go on dividing and dividing and dividing, and it will form a tumor. Okay. Now these tumors are of two types. One is a benign tumor, which is not dangerous, it is not cancerous, it will be where it is only. Okay. But the other one is a malignant tumor where it will spread. So this quality of spreading is what we call as metastasis. Okay, so through the process of metastasis, it's going to break off from one place and go. So I have a cancer tumor here, but these cells from my hand can go to my legs, can go to my stomach, can go to my brain, can go anywhere in the system. Okay, so as a result of which as the stages of cancer will prolong, if it is stage 4 and all, it's pretty much like I have no clue where any of these cells can be. So there can be multiple tumors, multiple places affected with cancer, and I cannot cure this person at all. Okay, and the person might die very soon. Okay, now if you look at the cause for cancer, it can be a chemical carcinogen, it can be a physical carcinogen like radiations, it can also be biological carcinogens like certain viruses that will activate, or certain factors that can activate the genes in our body. All of us have oncogenes, all of us have cancerous genes, but just that they are switched off. If you do things, if you eat too much of carcinogenic chemicals in your food, if you are exposed to too much of these carcinogens and you activate these genes, obviously you will get cancer. Okay, so it's not like one person will get cancer and the other person will not. Everybody has these genes, so it's up to you to protect yourself from activating these genes. Okay, is that clear to all of you? Perfect. Yes, there are a lot of viral oncogenes also, viruses that can transfer these oncogenes into us. Yes, Tarun. Okay, that's why I said viruses can cause, some diseases can cause, your exposure to different, this thing can turn on your oncogenes. So anything of it is possible. Okay, now if you look at the diagnosis, first thing is I can do a biopsy, okay, or I can do MRIs or CT scans or PET scans to see where the tumor is and how far it's spread and all that. I can check it out, and majorly through biopsy I can check if this cell is cancerous or not. Okay, and when it comes to treatment, I definitely have surgery. Now, if I've seen a lump, I definitely have to remove it. But along with surgery, I can do radiotherapy. Okay, so radiation therapy can be done where I will radiate the cells and kill these cells with harmful radiation. Or I can do chemotherapy, send drugs. Okay, or I can also do immunotherapy. I can do combination of these. Okay, a little bit of immunotherapy, a little bit of chemotherapy along with surgery, or however I can always play around it. Understood? That's all about cancer. Now, the last part of this particular chapter is the drugs. Okay, so the drugs, I will show you a simple table that you remember. Okay, we have opioids. Okay, then we have cannabinoids. Okay, and then last we have is coca alkaloids. Okay, now these opioids, where do they find, why do you find them? You find them in opium plant, which is called as Papaver somniferum. Okay, cannabinoids are called as Cannabis sativa plant. Okay, then coca alkaloids come from spelling mistake. Okay, erythroxylum coca. Okay, now what these opioids do, they bind to the CNS and gastrointestinal tract receptors. They will slow down the body and act like a depressant. Okay, they act like a depressant. Now, if you look at cannabinoids, what do? They bind to receptors in the brain. And same thing, they activate functions and make you feel like you have a sense of well-being, like a sense of euphoria. Now, cocaine or coca alkaloids basically do the same thing. They increase energy, okay, and they give a feeling of euphoria. Okay, now all of these drugs are temporary, but when they are overused, they can cause hallucinations, they can cause coma also in very extreme conditions. Okay, now what happens is majorly your body gets into to situations like withdrawal symptoms, which is when suddenly if you want to stop it, your body does not want to, because it has created a dependency on this drug. I cannot live without this drug, and that is what happens in addiction, which is nothing but a psychological, it's nothing but a psychological kind of attachment to that particular product. Okay, so it's very important to keep a check on your peers to see if any of them might be into such kind of abuse of drugs. Now, it is not definitely good for anybody, but as a society, as more people become dependent and addicted on drugs, it is very difficult to build a very healthy society and build a healthy thought process moving forward. Okay, so that's it, and we are done with the whole human health and disease chapter. Okay, now we only have one more chapter, which is microbes in human welfare. Only that one chapter is left. Okay, and this one chapter I'm going to take it in tomorrow's class. Okay, because I'm going to do a very small unit tomorrow, and I would like to complete this chapter tomorrow itself. Okay, so tomorrow we will finish cell. Okay, cell cycle, cell division, cell structure. We'll finish biomolecules as well as this microbes chapter tomorrow. Okay, is that clear? So same 4 p.m. I'll be taking the class tomorrow. So far we have completed 4 chapters in today's class. Okay, so I'll be seeing you all tomorrow at 4 p.m. and at 4 o'clock we will finish entire cell, biomolecule, cell division as well as the microbes chapter also. Okay, I'll finish for you in tomorrow's class. Is that clear, everybody? Pakka, Tarun, Humaira, everyone, is that clear? So don't forget to subscribe the channel and stay tuned. Tomorrow 4 o'clock I will be coming. Yes, 27 NIC plant physiology is coming, so I thought I'll club plant physiology with anatomy and morphology as well. So we'll finish all that on another day. Okay, everybody, that's it for today. So take care, everyone. I will see you all in the next class tomorrow at 4 p.m. Okay? See you all then. Take care. Bye-bye.